Number 1. Materpiscis attenboroughi, the 380 million-year-old fish fossilized mid-birth. Picture a shallow tropical reef on the northwestern edge of what is now Australia, roughly 380 million years ago. The continent we recognize today did not exist in that arrangement. The Gogo Formation of Western Australia was, in that distant moment, a thriving carbonate platform teeming with armored fishes, primitive sharks, and reef-building organisms. The Devonian period earned a nickname from later scientists. They called it the Age of Fishes. Jaws had only recently evolved. Coral reefs were experimenting with their earliest large-scale architecture. And swimming through those waters was an animal whose preserved body would, more than 300 million years later, rewrite the textbook on vertebrate reproduction. That animal was Materpiscis attenboroughi, a teectodontid placoderm. Placoderms were armored-jawed fishes, but they were not evolutionary failures or primitive dead ends. They were the dominant vertebrates of their time, equipped with bony plates covering the head and forward body. For decades, placoderms were portrayed as crude prototypes for later fishes. That portrayal collapsed when the Gogo material began to reveal what these animals were actually doing with their bodies. The specimen that changed everything contained two skeletons inside one. A mother, intact and articulated, carried an unborn embryo within her body cavity. Connecting the two was a mineralized structure interpreted as a fossilized umbilical cord. Soft reproductive anatomy almost never survives in the fossil record. Tissue decays. Bacteria consume cell walls within hours. For a placental connection to fossilize, conditions must accelerate mineral replacement before decomposition completes. Gogo preservation provided exactly those conditions. The fossils sit inside limestone nodules that protect three-dimensional anatomy. Paleontologists dissolve the surrounding limestone using dilute acetic acid. The bone, which resists the acid, emerges intact. The technique is slow and patient. Acid preparation can take months or even years for a single specimen, but it reveals fragile features that mechanical removal would destroy. John Long and his colleagues published the discovery in the journal Nature in 2008. Their analysis pushed the origin of vertebrate live birth back by approximately 200 million years. Before this fossil, the oldest accepted evidence for viviparity in vertebrates came from much later marine reptiles. Materpiscis collapsed that timeline. Internal fertilization implies copulation. That conclusion sits on additional anatomical evidence. Male placoderms possessed pelvic claspers, structures comparable in function to those seen in modern sharks. These appendages transfer sperm during mating contact. Before this work, the existence of placoderm sex was a quiet inference. Now it carries the weight of preserved evidence. The species name honors Sir David Attenborough. He had highlighted placoderms in his 1979 series Life on Earth. To call the find a filmed birth would overstate the situation. What we have is the oldest known vertebrate pregnancy frozen in stone. Comparing the embryo with modern live-bearing fishes provides a useful anchor. Its proportions are consistent with the late stages of internal development. The lesson sits beneath the headline. Sex, internal fertilization, and parental investment are not recent inventions. They are deep evolutionary strategies refined through hundreds of millions of years. The motherfish preserved behavior, not just bones, and that distinction matters when reconstructing the lives of extinct animals. If a fish could preserve the act of giving birth, what about an insect captured in the act of hunting? Number 2. The hell ants with metal-reinforced horns in Burmese amber. Step inside a droplet of resin trapped in time. Roughly 99 million years ago, a humid forest covered what is now northern Myanmar. The trees produced enormous quantities of sticky resin. Insects, mites, small reptiles, even feathers from non-avian dinosaurs got trapped, sealed, and eventually fossilized into amber. The Cretaceous forests of Burma left one of the richest insect archives ever discovered. Among the trapped insects were animals that looked, by every reasonable measure, fictional. Scientists call them hell ants. The formal name for the group is Markmitchelli. They are an extinct subfamily that flourished alongside the earliest modern ant lineages, then disappeared entirely. Modern ants are everywhere. Hell ants are nowhere. We only know them from amber. A modern ant's mandibles close from side to side, like a pair of pincers swinging horizontally. Hell ant mandibles did something else entirely. They rotated upward. Imagine jaws that swing vertically, like the blades of a bear trap mounted to the underside of the head. Now add a horn projecting from the top of the face. Prey approaching the ant would be pinned between the upward snapping jaws and the rigid horn above. The face was a trap. The trigger was contact. Philip Barden, working with Vincent Perichot and David Grimaldi, formally described one of the most striking species in 2017. They named it Lingua myrmex vladi. The name choice was deliberate. Vlad referenced the Dracula legend fitting an animal that may have impaled prey on a face-mounted spike. Other Haemimyrmecine ants, including species in the genus Haemimyrmex, share the upward jaw architecture but differ in horn shape and proportions. The horn itself produced one of the more remarkable surprises. Researchers identified biomineralized particles enriched with metals, particularly iron, embedded in the horn structure. Calling this metal reinforcement is accurate but easily misread. Nothing was forged. No metal sheet was hammered onto the animal. Instead, the ant's own biology deposited iron-bearing minerals into the horn, likely strengthening the surface against repeated impact. Other insects use similar strategies. Some beetles incorporate zinc or manganese into their mandibles. The hell ant pushed that same chemistry into a defensive prey-trapping rod. Amber preserves these details with extraordinary clarity. Resin captures three-dimensional cuticle, microscopic hairs, and the curvature of antennae. Some specimens preserve the moment of struggle. One famous fossil shows a hell ant gripping an immature cockroach relative, jaws locked into position, frozen during the kill. No modern ant uses a vertical jaw hunting system. Convergent evolution sometimes recreates similar tools, but the hell ant architecture is genuinely lost. These animals existed in a Cretaceous experiment with social insect predation, then vanished. Their tiny scale, often only a few millimeters long, makes the anatomy more striking, not less. Nature produced a predator that looks invented, then sealed it inside a transparent tomb. The amber itself becomes the witness. If insects can be preserved during the act of hunting, what happens when an entire underground tunnel system becomes the fossil? Number 3. The Devil's Corkscrews of Nebraska, also known as Daemonilics, stand in the Badlands of western Nebraska in the late 1800s. 8-foot-tall spiral structures rise from the eroding sediment. Their surfaces are smooth, their twists are nearly perfect, they look engineered. Early geologists who encountered them assumed they could not be natural. Yet there they were, hundreds of them, twisting upward from the high plain soils. The name Daemonilics, sometimes spelled Demonilix, means devil's corkscrew. It captured the unease early observers felt. The structures occur in the Harrison Formation, deposited roughly 22 million years ago during the Miocene epoch. The American Midwest at that time was a transitional landscape. Forests were retreating, grasslands were expanding. Animals had to adapt to drier conditions, more open visibility, and seasonal extremes. Theories piled up quickly. Some researchers proposed the spirals were giant freshwater sponges. Others suggested fossilized plant roots from extinct trees. A few argued they might be unusual geological structures formed by groundwater dissolution. None of those theories explained the consistent helical geometry or the tooth marks etched along the inner walls. The breakthrough came with a category distinction. Daemonilics is not a body fossil. It is a trace fossil. Trace fossils preserve behavior, activity, or environmental modification rather than the body of an organism. A footprint is a trace fossil. A burrow is a trace fossil. So is a corkscrew tunnel dug by an animal that no longer exists. The maker turned out to be Palaeocastor, an extinct land beaver. The modern beaver builds dams and rivers and chews through trees. Palaeocastor did neither. It was a small terrestrial rodent similar in some ways to a prairie dog that dug deep helical burrows into firm sediment. The spiral led downward, ending in a flatter living chamber where the animal sheltered, raised young, and waited out the worst of the climate. The tooth marks visible inside Daemonilics burrows became the key piece of evidence. Their spacing matched the incisors of Palaeocastor. Better still, complete skeletons of Palaeocastor turned up at the bases of some burrows. The animal had died inside its own architecture, leaving the maker physically connected to the structure. Larry Martin and Deborah Bennett resolved the puzzle in 1977. Their analysis tied the corkscrews directly to Palaeocastor behavior and established Daemonilics as one of the clearest examples of preserved animal architecture in the fossil record. The helix shape may have helped with several practical problems. It could stabilize tunnel walls against collapse. It might regulate temperature and humidity, since the deeper chambers remained insulated against surface heat and cold. And the spiral shape probably slowed predator access by lengthening the path of attack. The Miocene grasslands rewarded burrowers. Open habitats meant fewer trees but easier sightlines for predators. Going underground became survival. Paleocastor was not building dams, it was engineering screws into the earth. If a beaver could leave a fossilized blueprint of its home, what mark might a much older human relative have left on a much smaller object? Number 4. The 540,000-year-old shell engraved by Homo erectus. Travel to a museum drawer in the Netherlands. Inside that drawer rests a freshwater mussel shell collected in the 1890s from the Trinil site in Java, Indonesia. The same site produced the original fossils of Homo erectus, the species sometimes called Java man. For more than 100 years, the shell sat in storage. It had been catalogued, examined, and returned to its drawer. Nobody noticed what it carried. In 2014, Josephine Jordaens and her colleagues at Leiden University published a reanalysis in the journal Nature. The shell, identified as a pseudodon mussel, carried a deliberate zigzag engraving along its outer surface. The lines were not random scratches. They formed a structured geometric pattern cut with consistent depth and intention. The dating placed the engraving between approximately 540,000 and 430,000 years old. That range falls deep into the time of Homo erectus, long before the earliest accepted symbolic engravings made by anatomically modern Homo sapiens. The conventional assumption had been that abstract mark-making belonged to our species, perhaps reaching back to roughly 100,000 years ago in southern Africa. The Trinil shell pushed possible deliberate geometric engraving back fivefold. The word art is tempting here, but scientists use it carefully. Calling the marks art implies symbolic intent that cannot be confirmed from a single object. The safer language is deliberate geometric engraving by an archaic human. Microscopic analysis showed that the grooves were incised rather than abraded. The cross-sections matched cuts made by a sharp point, and the directionality indicated a controlled hand pulling a tool through the shell surface. The probable tool was a shark tooth, which trinial sediments preserve in abundance. The shell also showed evidence of opening. A small hole had been pierced near the adductor muscle, the muscle that holds the two valves shut. Punching through that muscle would have caused the shell to relax open, providing access to the meat inside. Pieced together, the evidence suggests Homo erectus was exploiting freshwater shellfish with tools, opening shells in a controlled way, and modifying their surfaces. Another shell from the same collection had been worked into a tool. Its edge showed wear patterns consistent with cutting or scraping. Shellfish were not just a food source, they were also a raw material. Freshwater shellfish matter for hominin diet for a specific reason. They concentrate certain fatty acids and trace minerals that support brain development. Regular consumption of shellfish may have contributed to the cognitive trajectory of our genus across the Middle Pleistocene. The zigzag itself remains undeciphered. Whether it meant anything is unknown. The pattern may have been decoration, identification, practice, or pure expression. The point is that the behavior of structured mark-making did not begin with us. 2 excavations made the discovery, the original one in the 1890s and the reanalysis more than a century later. The object never moved. The way we looked at it changed. If a marking on a shell can quietly rewrite human prehistory, what might an entire drowned village look like when it finally surfaces? Number 5. The underwater megalithic circle of Atlit-Yam. Drop beneath the Mediterranean Sea off the Carmel coast of Israel. At depths between 8 and 12 meters, divers move through the remains of a Neolithic village. The site is called Atlit-Yam. It is approximately 9,000 years old, dating to roughly the 7th millennium BCE. It sat on dry land when it was occupied. It now lies underwater because global sea levels rose dramatically after the last ice age. Ehud Galili, working with the Israel Antiquities Authority, began systematic underwater investigations of Atlit-Yam in the 1980s. The work continues today. The site preserves stone architecture, organic remains, human burials, and behaviors that would not have survived on a dry, exposed land surface. Salt water and sediment slowed bacterial decay. Wave energy at the depth of the site is modest, which limits erosion. The inhabitants were not a single economy community. They farmed cereals, herded sheep, cattle, and goats, fished offshore, and gathered marine resources. The settlement reflected a Neolithic transition in motion, with people balancing several food strategies at once. A central feature of the site is a megalithic installation. Around or near a freshwater spring stand 7 stones, each weighing roughly half a ton, arranged in a deliberate pattern. Some of the stones carry cup marks, small, carved depressions on their upper surfaces. Cup marks recur in Neolithic and Bronze Age sites across Eurasia, often associated with ritual or water-related practices. Freshwater springs were not just utilitarian features in early communities, they were focal points of survival, gathering, and ceremony. The site also includes stone-lined wells, among the earliest known examples in the region. Wells reveal how communities solved water access problems when surface springs began to fail. The fact that some Atlit-Yam wells contain fill consistent with abandonment debris suggests that water quality eventually deteriorated. As sea levels rose, saltwater intrusion could have contaminated freshwater aquifers, undermining the village's water supply long before the buildings themselves drowned. Human burials within the settlement provided another dimension of evidence. Israel Hershkovitz and his colleagues confirmed ancient tuberculosis in remains from the site. The disease was identified through ancient DNA and lipid biomarkers preserved in the bones, including those of a woman and a child buried together. Tuberculosis is associated with settled life, animal husbandry, and population density. Atlit-Yam shows the disease at an early stage of that transition, providing a direct molecular link between Neolithic lifestyle change and the diseases that came with it. Comparisons with the Atlantis legend are tempting and unproductive. Atlit-Yam is grounded firmly in archaeology. The site is dated, mapped, and studied with mainstream methods. Its preservation underwater is what makes it strange, not its origin. Rising seas drowned an ordinary Neolithic community, and the ordinary became extraordinary because the sea protected what time would have erased on land. If water can preserve a whole village, what happens when prehistoric bones get replaced by something far more valuable than stone? Number 6. The opalized prehistoric creatures of Coober Pedy. Moved to the South Australian outback to Coober Pedy. The town sits in one of the harshest landscapes in the country, but it produces the majority of the world's precious opal. Miners dig shafts into the rock searching for the gemstone. Sometimes they encounter something else entirely. They find fossils. The fossils are themselves opal, formed by the same geochemical process that produces the gems. The most famous specimen is known by the nickname Eric. Eric is not, despite popular labeling, a dinosaur. The skeleton belongs to Umunosaurus democillus, a small, short-necked marine reptile related to plesiosaurs. The fossil came out of a Coober Pedy opal mine in 1987. A private collector acquired it. In 1993, the Australian Opal Centre and the Australian Museum, supported by a public fundraising campaign, raised the funds to bring Eric into public scientific care. Eric's bones look like polished jewelry because they are. During the Cretaceous period, a vast inland sea covered much of Australia. Marine reptiles, fish, and ammonites swam through those waters. When they died, their remains sank into the sea floor. Silica-rich groundwater later moved through the sediment, dissolving original bone material and replacing it with opaline silica. In some cases, only the cavities filled with opal. In others, the entire bone became opal. The result is fossil material that flashes red, blue, and green under light. Microcomputed tomography work on Eric revealed preserved gut contents. Fish bones inside the abdominal region had themselves opalized. The same scans recovered gastroliths, small swallowed stones found in the digestive tract. Gastroliths show up in many marine reptiles. Some scientists argue they aided digestion by grinding food. Others suggest they functioned as ballast, helping the animal control buoyancy in the water column. The debate continues, but the presence of gastroliths inside Eric provides direct evidence of behavior alongside anatomy. Australia is globally unique in the abundance and quality of its opalized fossils. True opalized dinosaur remains are better known from Lightning Ridge in New South Wales, where partial skeletons of small ornithopod and theropod dinosaurs have surfaced from opal fields. Coober Pedy and Lightning Ridge together represent a rare convergence of paleontology and gemstone geology. The convergence carries a cost. Miners are not paleontologists. Their primary interest is selling opal. A scientifically priceless fossil can easily be cut and sold as jewelry before any scientist ever sees it. Some specimens have likely been lost to the gem trade entirely, broken up and dispersed across collectors. Programs that connect miners with paleontologists, including public buyback initiatives, try to keep important finds in scientific hands. The beauty of these fossils is not separate from their data. The same chemistry that produced the gemstone preserved bone shape, gut contents, and small anatomical structures. Eric is a sea reptile, a fossil, and a piece of natural treasure simultaneously. If groundwater can replace ancient bone with opal, what can salt marsh peat do to a wooden structure built by a much more recent culture? Number 7. Seahenge. The Bronze Age circle built around an upside-down tree. Walk along the Norfolk coast of eastern England near the village of Homo next to the sea. At low tide in 1998, the receding water revealed a ring of dark timber posts buried in the beach. A local beachcomber spotted the central feature first, a large upside-down tree stump. The roots pointed up toward the sky. The trunk pointed down into the sand. Around the stump stood 55 split oak posts, arranged in a tight circle. The site was named Seahenge. Its formal designation is Home 1. Dendrochronology, the science of tree ring dating, anchored the structure to a precise date. The oaks used in the monument were felled in the spring or early summer of 2049 BCE. That precision is rare in archaeology. Most prehistoric dates carry margins of several centuries. Dendrochronology compresses that margin to a single season of a single year. The narrow seasonal window matters. It means a coordinated work effort within weeks. Bronze Age communities cut large oaks, split them, and transported them to the site within a planned, organized timeline. The number of timbers required a labor force, planning, and shared purpose. The wood preserved axe marks. Some axes left distinctive cutting patterns that allowed researchers to identify the work of individual tools and, by inference, individual workers. Up to 50 axes were used in the construction, suggesting a community-wide project. The carpentry was skilled. Split halves of trees were placed bark side outward, forming a closed timber wall around the central feature. Seahenge originally stood in salt marsh, not on open beach. The surrounding peat and sediment buried the lower portions of the timbers, sealing them in a waterlogged, low-oxygen environment. Peat is hostile to bacterial decay. Wood that would have rotted on dry land survived for nearly 4,000 years because it never had a chance to dry out and never received the oxygen bacteria need to consume it. In the late 20th century, coastal erosion stripped away the protective peat. The monument emerged from its grave on the beach. The exposure was rapid, dramatic, and dangerous. Wind, tides, and biological activity began damaging the timbers immediately. English Heritage authorized excavation in 1999. The decision to remove the timbers was controversial. Some local groups objected on spiritual and heritage grounds. Archaeologists argued that leaving the timbers in place would mean watching them disintegrate. The timbers were lifted, conserved through years of preservation treatment, and eventually placed on display at the Lynn Museum in King's Lynn. Interpretations of Seahenge vary. The most widely cited proposal connects it to mortuary ritual. The inverted oak may have functioned as a platform for excarnation, the practice of exposing the dead to allow flesh to be removed before secondary burial. Inversion may have symbolized a passage between worlds. Other interpretations emphasize ritual enclosure, ceremonial use, or seasonal ceremony tied to the spring felling date. None of the theories have been confirmed. The function remains a question, even though the timing is now known to the season. If wet peat can preserve a Bronze Age monument, can the same chemistry preserve something far softer, far stranger, and far less likely to survive at all? Number 8. The Heslington Brain. In 2008, construction crews working at the University of York's Heslington East campus uncovered an Iron Age skull buried in waterlogged mud. The remains date to roughly 673 to 482 BCE. The skull was that of an adult male. The rest of the body was missing. Inside the skull was something nobody expected. Brain tissue, approximately 2,500 years old, still folded, still recognizably structured. The brain had survived. The rest of the soft body had not. Sonia O'Connor and her colleagues published the analysis in 2011. They examined the surviving tissue using microscopy, chemistry, and comparative anatomy. The external morphology was preserved. The shrunken organs still showed gyri, the characteristic ridges on the surface of the brain. Histology was limited because the internal cellular structure had been altered by time. Claiming preserved neurons would overstate the evidence. What survived was the gross structure, fixed in place by chemical changes within the tissue itself. The man had died violently. Evidence on the neck vertebrae indicated hanging or some other form of fatal trauma to the neck. The head was then separated from the body. The skull was buried in wet, low-oxygen sediment shortly after death. Brain tissue normally decays within days. The process begins with autolysis, the self-digestion of cells by their own enzymes once a cellular regulation breaks down. Bacterial decomposition follows, with gut bacteria entering tissues and consuming organic material. Decapitation may have shortened the chain. By separating the head from the body, it isolated the brain from the major bacterial sources lower in the trunk. Burial in anoxic mud removed the oxygen needed for further decay. Acidic conditions slowed surviving bacterial activity. Later research focused on protein chemistry. Folded protein aggregates within the brain tissue appear to have stabilized the structure. Some proteins, when exposed to specific conditions, form tightly folded clumps that resist enzymatic breakdown. These aggregates may have provided a kind of internal scaffold, preventing the tissue from collapsing into the chemical pulp expected after thousands of years. The Heslington brain is not the only preserved ancient brain, but it remains one of the best studied. Its survival depends on a sequence of unlikely events. The man had to be killed in a way that separated the head from the body. The head had to be deposited rapidly in a specific kind of waterlogged sediment. The chemistry of that sediment had to support protein stabilization rather than tissue breakdown. The face, flesh, and skin were lost. The most fragile organ in the body remained. That inversion of expected preservation is the strangest feature of the find. Soft tissues usually disappear first. The brain usually disappears earliest of all. At Heslington, the order ran backwards. If a brain can persist when everything else fails, what about a skull whose entire history was suppressed for almost a century? Number 9. The Dragon Man's skull, hidden in a well for 85 years. The story begins during bridge construction in Harbin, northeastern China, in 1933. A laborer discovered an unusual fossil cranium while working on a bridge across the Songhua River. Japan was occupying parts of northeastern China at the time, and the worker did not turn the find over to authorities. Instead, he wrapped it and hid it at the bottom of a family well. The skull stayed in that well for the next 85 years. Only on his deathbed did he tell his family what he had hidden. The cranium reached scientists in 2018. In 2021, Chang Ji, Sijun Ni, and their colleagues published a description of the skull. They named the proposed new species Homo longi, where longi derives from Heilongjiang, the province where Harbin sits. The river name translates roughly as Black Dragon, which gave the specimen its informal nickname, Dragon Man. The cranium is at least 146,000 years old, placing it in the late Middle Pleistocene. It is massive. The brow ridges are heavy, the face is broad, the brain case is large, with an estimated brain volume comfortably within the range of Neanderthals and modern humans. The combination of features does not match any previously named species cleanly. Taxonomic placement remains contested. Some researchers accept Homo longi as a distinct species. Others argue the skull may represent a regional variant of an already known group. The most consequential debate centers on a different question. Could Homo longi be a Denisovan? Denisovans are an extinct human group known primarily from fragmentary remains and ancient DNA. The original Denisova cave finds in Siberia consisted mostly of a finger bone, teeth, and a fragment of jaw. Researchers established the group's existence largely through molecular evidence. What Denisovans actually looked like, particularly their faces, has remained a question for over a decade. In 2025, molecular evidence linked the Harbin cranium to the Denisovan lineage. Ancient proteins recovered from the bone suggested Denisovan affinity. Mitochondrial DNA extracted from dental calculus, the hardened plaque on the teeth, reinforced the connection. If those results hold, Homo longi may finally provide a face for the previously faceless Denisovan group. A weakness remains. The provenance of the skull is uncertain. The original sediment layer is unknown because the find was hidden for so long. Without the original geological context, some details of dating and association must be inferred from chemistry rather than stratigraphy. Critics emphasize this gap. Supporters argue that direct dating of the bone and consistent molecular results compensate for the missing field context. A skull hidden in a well for the better part of a century has become a possible answer to one of the largest open questions in recent human evolutionary research. The face of a ghost lineage may have emerged from a private secret. If a hidden skull can change human ancestry, what does it take to preserve an entire armored dinosaur, as if it fell asleep yesterday? Number 10. Borealopelta markmitchelli. The dinosaur preserved so perfectly, it looks asleep. In 2011, a shovel operator at the Suncor Millennium Mine in Alberta, Canada, noticed something unusual in the oil sands he was excavating. He stopped digging. The Royal Tyrrell Museum of Paleontology sent recovery specialists. What emerged from the rock was an armored dinosaur, preserved with skin, scales, armor plates, and even the keratinous sheaths that once covered its horns. The animal is Borealopelta markmitchelli, a notosaurid ankylosaur. It lived approximately 110 to 112 million years ago in the early Cretaceous. The species name honors Mark Mitchell, the Royal Tyrrell technician who spent more than 7,000 hours preparing the fossil from the surrounding rock. The formal description was published in 2017 by Caleb Brown and colleagues. Notosaurs were heavily armored, herbivorous dinosaurs that lack the bony tail club seen in their cousins, the ankylosaurids. They relied on body armor and possibly on bright displays for predator avoidance. Borealopelta was about 5.5 meters long and weighed approximately 1.5 tons during life. The fossil is terrestrial in origin, but the carcass ended up at sea. After death, the body apparently floated, then sank, then settled into the soft mud of a Cretaceous seabed. Rapid burial prevented scavenging and slowed decay. The mud sealed in three-dimensional anatomy. Most dinosaur fossils are flattened, crushed into 2 dimensions by overlying sediment. Borealopelta retained its body shape. The animal sits on display today looking like a sleeping armored reptile, not a skeleton. The preservation extends beyond shape. Chemical analysis recovered evidence of pigmentation. Reddish-brown coloration appears to have covered the upper body. The underside was lighter. This pattern is called countershading, a form of camouflage used by many modern animals from sharks to deer. Darker top surfaces match the look of a viewer looking down from above. Lighter undersides match the brightness of a viewer looking up from below. Countershading on a 1.5-ton armored herbivore suggests that, even with extensive bony defenses, Borealopelta faced predation pressure significant enough to favor visual camouflage. The predators of its time were large enough to threaten an armored adult. The animal's last meal survived. Stomach contents inside the body cavity were dominated by fern material, with smaller amounts of other plant tissue, charcoal fragments, and small stones likely swallowed during feeding. The charcoal suggests the dinosaur fed in an environment that had recently burned, possibly during a fire-disturbed phase of forest regrowth. The fern dominance helps reconstruct the surrounding plant community. The combination provides direct dietary data unmediated by inference from tooth shape. Borealopelta is one of the most complete and informative dinosaur fossils ever recovered. Skin, color, armor, meal, and shape are preserved together. The fossil reads less like bones and more like a body. A sleeping dragon, frozen in stone, brought back to view by industrial mining and 7,000 hours of patient preparation. 10 extraordinary discoveries share a common quality, but the reasons they survived are not the same. What makes the difference between a body that lasts and one that vanishes? The discoveries covered so far all sound, on first hearing, slightly impossible. Live-bearing fossil fish, hell ants in amber, fossilized burrows, engraved shells, underwater villages, opal dinosaurs, drowned timber circles, ancient brains, hidden skulls, sleeping armored reptiles. Each of them survived because nature accidentally built a vault around the right material at the right time. Limestone nodules in the Gogo Formation worked as miniature stone safes. The carbonate chemistry encased the placoderm bodies, protecting fragile structures, including the umbilical connection of Materpiscis. Acid preparation slowly dissolved the limestone, leaving the bone intact. Mineralization can take three forms: replacement, where original material is swapped for minerals atom by atom; coating, where minerals form layers around a structure; and infilling, where cavities fill with mineral material. Go-go preservation combined all three. Amber works differently. Resin captures organisms in soft form, then hardens around them. The cuticle of an insect, including microscopic hairs and metal-rich horn deposits, can survive nearly intact. Hell ants in Burmese amber illustrate the principle. The resin sealed them so completely that even the moment of predation was frozen. Deinonychus preserves no body and represents a different category entirely. Trace fossils preserved behavior. The burrow is not the beaver. It is the record of the beaver's work. Behavior fossilizes when an animal modifies its environment in ways that the environment then retains. Trinil shells last because shell is durable. Calcium carbonate resists decay if conditions stay reasonably stable. Engraved surfaces, once cut, preserve the geometry of the cuts. Shell becomes an archive of tool marks because the substrate is hard enough to record them and resistant enough to keep them. Atlit-Yam represents underwater burial. The Mediterranean does not behave like the open ocean at that location. Sediment built up around the stone structures. Wave energy stayed low. Saltwater inhibited bacterial decay of certain materials while allowing preservation of others. Submerged archaeology offers both protection and threat. Protection from looters and exposure, threat from currents, marine organisms, and gradual erosion. Coober Pedy fossils involve silica-rich groundwater. Dissolved silica moved through buried sediment. Where bone cavities existed, the silica precipitated into opal. The same chemistry that produces gemstones produced fossils. Opalized bone differs from ordinary permineralized bone in the specific mineral that fills the structure. Both record shape, only opal flashes color. Seahenge survived in peat. Waterlogged organic rich sediment excludes oxygen. Bacteria that need oxygen cannot function. Wood that would rot in open air persists for thousands of years. Once the peat eroded away, the survival window closed quickly. The site had to be excavated immediately or lost. The Heslington brain occupied a similar chemical environment, anoxic mud combined with proteins that aggregated rather than dissolved. The biochemistry of protein folding became part of the preservation pathway. Brain tissue is normally the first organ to vanish. Under the right anoxic chemistry with the right protein behavior, it can outlast the rest of the body. The Harbin skull preserves well in itself but lost its original context. Bones can survive in many environments. What fails when a fossil is hidden is the stratigraphic information that surrounds it. The skull is real. Its layer is not certain. Borealopelta required rapid marine burial of a land animal. The combination is rare. Terrestrial animals usually decay on land or are scavenged. Marine burial of a land carcass demands the body to reach the sea, sink, and be covered with sediment fast enough to seal in soft structures. Each step shaves off the odds. Soft tissues need speed, specific chemistry, or isolation to survive. Hard tissues are more forgiving. Behavior survives when an animal alters durable matter. Preservation is not random luck alone. It is a layered process where geology, chemistry, and biology cooperate in narrow, identifiable ways. If preservation explains why these objects exist, what explains how they reach scientists in the first place? Most major discoveries do not begin in universities. They begin with miners, divers, beachcombers, bridge workers, farmers, and shovel operators. The path from discovery to scientific description usually passes through several human hands. Coober Pedy miners encounter fossils as a byproduct of opal extraction. The fossils are often beautiful enough to sell. Some specimens vanish into private gemstone markets before any paleontologist sees them. Buyback programs and partnerships between museums and miners try to redirect significant finds into scientific hands. The Suncor shovel operator who paused at Borealopelta represents a different model. Industrial operations sometimes recognize value beyond their immediate work. The Royal Tyrrell Museum maintained relationships with the mining industry that supported rapid response. Without those relationships, the specimen might have been damaged or processed before recovery was possible. Seahenge surface because a beachcomber walked the right stretch of Norfolk coast at low tide. The same erosion that exposed the timbers threatened to destroy them. Tides reveal and damage in the same motion. Coastal archaeology often operates on short time horizons because exposure rarely lasts. The Harbin skull spent 85 years hidden in a family well. War, occupation, and personal memory shaped its trajectory more than any institution. Hidden provenance creates lasting problems. The skull arrived without sediment, without associated material, and without records of the original find spot. Science can still extract enormous information from the object itself, but some questions can never be answered with the original layer missing. The Trinil shell illustrates a different category. The shell was excavated in the 1890s as part of a documented collection. It then sat in storage for over a century. Reanalysis became the second act of discovery. The technology, microscopy, and dating methods needed to read the engraving did not exist when the shell was first collected. Museum collections constitute a vast reservoir of unrealized discoveries waiting for new methods. Gogo fossils require fieldwork plus extended laboratory preparation. The acid preparation that reveals Materpiscis can take years per specimen. The discovery in the field is only the first stage. The discovery in the lab follows. Atlit-Yam demands underwater archaeology. Divers, mapping equipment, sediment cores, and careful context recording are required. The pace is slower than terrestrial digs. The information is more fragile. Salt and currents constantly modify the site. Heslington emerged through construction archaeology. The University of York was expanding its campus. Required archaeological surveys before construction revealed the Iron Age remains. Routine planning regulations, rather than research priorities, brought the brain to scientific attention. Spectacular appearance is not the same as documentation. Scientists insist on chain of custody records, geological context, and methodological reproducibility. Fake-sounding discoveries become credible through that documentation, not through their visual impact. Science, in the end, is recovery, preparation, verification, and preservation. None of those steps can be skipped without weakening what is learned. If discovery brings objects to laboratories, what specific techniques convert them from curiosities into confirmed evidence? The first question every extraordinary find faces is how anyone knows it is real. The answer comes through layered techniques that cross-check each other. Radiometric dating uses the decay of radioactive isotopes to assign numerical ages. Where volcanic ash layers occur near a find, methods like argon-argon dating provide reliable dates within known error margins. Where direct radiometric dating is not possible, biostratigraphy steps in. Biostratigraphy uses fossil assemblages within a rock layer to assign relative age based on the known time ranges of indicator species. Dendrochronology delivered the precise spring or summer felling date of Seahenge in 2049 BCE. The method matches tree ring patterns from one wooden specimen against established master chronologies. Where the match is robust, dating reaches annual precision. No other dating method available for the period offers that resolution. Microscopy proved that the Trinil shell engraving was deliberate. Cross-sections of the grooves revealed the geometry of cuts. Tool mark directionality indicated a controlled hand. Random scratches and biological scuffs leave different signatures from incised lines. The difference shows under magnification even when it remains invisible to the naked eye. Amber preservation supports comparative anatomy at extraordinary resolution. Hell ant jaws were reconstructed by directly observing the three-dimensional structures in amber, then comparing them with living and extinct insect lineages. Functional models followed from anatomical observation. CT scanning provides non-destructive imaging inside fossils. For opalized specimens like Eric, the technique is essential. Cutting into the fossil would destroy both the scientific specimen and the gemstone material. CT reveals internal anatomy, gut contents, and gastroliths without a single cut. Borealopelta supports chemical pigment analysis. Researchers identified residues consistent with melanosomes, the cellular structures that produce pigment in skin and feathers. Pigment chemistry must be interpreted cautiously, but the convergence of multiple chemical markers supports the counter-shading conclusion. Stomach contents in the same specimen verify diet directly. There is no inference from tooth shape required. The ferns, are in the body. Ancient DNA and lipid biomarkers confirmed tuberculosis at Atlit-Yam. The disease leaves specific molecular signatures in bone. Recovery of those signatures from a woman and child confirmed that tuberculosis was already present in human populations 9,000 years ago. Protein analysis stabilized the interpretation of the Heslington brain. Paleoproteomics, the study of ancient proteins, has emerged as a powerful tool over the past 15 years. For the Harbin skull, dental calculus DNA, combined with bone proteins, built the proposed link to Denisovans, calcified plaque on teeth traps, biological material from the individual and from their environment. It has become an unexpected reservoir of molecular evidence. No single method is sufficient on its own. Extraordinary finds rest on converging lines of evidence. Provenance gaps weaken but do not erase what specimens reveal. Where the chemistry, the anatomy, the dating, and the context all agree, claims become defensible. Where they disagree, scientists keep working. If the methods can verify the objects, what do experts actually argue about regarding their interpretation? Strange finds attract multiple explanations. Some explanations stay close to the evidence, some pull further away. Examining the competing views shows how the scientific community processes unusual material. The mainstream theory treats each discovery as a real object explained by rare preservation conditions and recoverable context. For Materpiscis, this view holds that the specimen represents the oldest known direct evidence of vertebrate live birth, with copulation implied by male placoderm claspers. For hell ants, the mainstream view sees an extinct predatory anatomy refined through millions of years of selection, not a fantasy exaggeration. For Dimonelics, the mainstream view established by Larry Martin and Debray Bennett in 1977 identifies the spirals as paleocastor burrows, with helical shape providing structural and thermal advantages. For the Trinal shell, the mainstream view accepts deliberate geometric engraving by Homo erectus while withholding the loaded term art. For Atlit-Yam, the mainstream view treats the village as a drowned Neolithic settlement, with sea level rise from post-ice age, warming as the cause of submersion. For opal fossils, the mainstream view explains the appearance through silica replacement in bone cavities. For Seahenge, the mainstream view sees a Bronze Age timber ritual or mortuary monument with mortuary use as the leading interpretation. For the Heslington brain, the mainstream view identifies a rare combination of anoxic burial, protein aggregation, and rapid decapitation as a preservation pathway. For Harbin, the mainstream view recognizes an archaic human with growing molecular evidence for Denisovan affinity. For Borealopelta, the mainstream view emphasizes rapid marine burial of a land carcass with counter-shading evidence for visual predator avoidance. An archaeological and historical theory adds a second layer. Human context shapes what survives and what disappears. Hidden wells, museum drawers, mining markets, and tides change discovery timelines and complicate provenance. What scholars sometimes call lost knowledge often turns out to be modern failure to recognize evidence already in storage. The Trinal shell embodies this point. Atlit-Yam and Seahenge demonstrate ritual behavior that we cannot fully reconstruct from material remains alone. The objects survive, the meanings often do not. A fringe or speculative theory exists for almost every find on this list. These interpretations connect discoveries to myths, lost civilizations, ancient technology, or non-human agency. Dimonelics has been called a fossilized machine. Seahenge has been described as a portal. The opal fossils have been linked to lost continents. Atlit-Yam draws Atlantis comparisons. Hell ants attract claims of intentional design. These interpretations carry cultural weight, but they sit outside the evidence base that scientific methods can test. Treating fringe interpretations as equal to the evidence-based mainstream would misrepresent the strength of the data. Treating them as worthless dismisses the real cultural function they perform. People reach for myth-shaped explanations when reality looks impossible. The discoveries in this list look impossible, and that is what produces the speculation in the first place. The deeper point is that the documented evidence is stranger than the speculation. A fossilized birth, a metal-strengthened horn, a corkscrew burrow, a brain that outlives a body, and an armored dinosaur with preserved color are already extraordinary. They survive scientific testing precisely because they are real, What do all 10 discoveries taken together say about deep time and the human attempt to read it? 10 discoveries connect through improbability. None of them require exaggeration to be remarkable. The fossil record and the archaeological record preserve more than species and objects. They preserve moments, decisions, and behaviors. Reproduction in Materpiscis links to predator design in hell ants. Both are biology preserved as action. Burrow engineering in Paleocastor links to mark making by Homo erectus. Both are behavior recorded in durable material. Drowned villages link to rising seas. Opalized bones link to mineral chemistry. Seahenge links to Bronze Age cosmology. The Heslington brain links to decay science. Harbin links to human ancestry. Borealopelta links to ecological reconstruction. Each of these connections survives because scientific methods convert wonder into evidence. Dating, microscopy, chemistry, and contextual analysis together produce conclusions that hold up under scrutiny. The phrase fully documented does not mean that every question is closed. It means that conclusions have been tested across multiple methods and remain defensible. Some meanings remain unreachable. The function of Seahenge cannot be fully reconstructed. The intent behind the trinal zigzag cannot be confirmed. The full life of a Homo longi individual cannot be recovered. Ritual and symbolic dimensions often resist the methods that work well on bone, sediment, and chemistry. Deep time does not need legends. The evidence is already unbelievable.